Our next speaker will be Ben Langmead from Johns Hopkins University. He's going to talk about alignment using prior knowledge.
Good. All right. Good morning. So I want to talk a little bit today about read alignment. And read alignment is the first step in a lot of genomic sequencing data analyses. It's been called, and as soon as I saw this, I wrote it down so I could include it in my talks. It's been called the cornerstone of modern genomics. And it's been studied for a long time, and you might think that we therefore know pretty much all there is to know about it, but we don't. And so some of the questions that I think are more or less still pending in the literature are questions like, how do we do a good job of predicting mapping quality, the probability that an alignment is incorrect? How do we characterize what the heuristics are actually missing when we do alignment heuristics? And what's an appropriate scoring scheme? And hat tip to John Kesasiaglu, who covered this his work with Dan de Blasio and others yesterday, which tackles this topic with respect to multiple alignment. What I want to talk about today is a particular sort of trend that I see where there are different variations on the alignment problem, not even very complicated or new variations on the alignment problem, but where the scoring schemes we typically use are not appropriate, and we sort of do a dance to get around the fact that the scores are not appropriate, and I'm advocating that we think about this a little bit more thoroughly. So typical read aligners use a pretty standard affine gap penalty style scoring scheme, as John was discussing yesterday. It consists of basically a kind of substitution matrix for the different nucleotides, and then a gap open and a gap extension penalty. So the penalty has an AX plus B type form, where there's an extension penalty that gets multiplied by the length of a gap when there's a gap in an alignment, and then there's a constant term that's always added whenever there's a gap in an alignment. And most of the commonly used tools have more or less comparable schemes. I have QU here to indicate that this depends on the base quality value. These are just mild variations on a theme. And I didn't want to show these without showing sort of the original, right? Classic PAM substitution matrix is a version of this as well. And so that's the typical scoring scheme used by these tools. On the other hand, and sort of side to side with the scoring scheme is its cousin, the heuristics, that are used in the actual alignment algorithm. And the typical heuristics are seed and extend, right? So, or variations on the theme of seed and extend. Seed and extend and affine gap penalty go fairly well together. And they've sort of evolved together. So they're basically a good match. Now, where does the scoring scheme start to sort of break down? Well, one of the places it starts to break down is in spliced alignment. So if you're aligning sequencing reads that come from mature messenger RNAs, as we do often with RNA-seq alignment, then some of those mature mRNAs are going to span an intron that's been spliced out. And in that case, when you go to try to align it back to the reference genome, you'll find that there's a gap in the alignment corresponding to the intron. And these gaps can be very long, right? This was discussed this morning, right? With recursive splicing, they're super duper long. So this is a case where the affine gap penalty is going to fall down a bit because it's expecting gaps that are A, relatively short, and B, it's not going to pick up on either of the two salient factors that I think are probably most relevant to scoring an intron, which is, does it have the appropriate splice motifs on either side of the intron? You may even look inside the intron for splice motifs. And does it have an appropriate length? And intron length distributions, we already saw plots of this earlier this morning. They tend to have modes, and then they fall off on either side. And sometimes they have multiple modes. And then you've got your recursive splicing introns probably like way out here. So the typical affine gap penalty scoring function doesn't know about this. So what do? This is another example of how it's species specific as well. So what do? We kind of split the alignment problem up into two phases, which will be a theme. So we split the alignment problem up into two phases. In the first phase, what tools do is they take the reads, they split them up into substrings. They could be overlapping substrings or not. I'll call these readlets. The readlets get aligned to a genome basically with standard scoring schemes, although they tend to be pretty short. So when I say standard scoring schemes, we're not allowing very much in terms of mismatches and gaps. And then when they when they align to the reference genome, they can fall in certain patterns that seem to be indicative of splice junctions. So for example, here, we might say, okay, these guys fell over here, these guys fell over here. We might look here and find a donor and an acceptor, and then become convinced that that must be a splice junction. So then we can take the things that we consider to be true splice junctions or true introns and actually put them in the reference genome representation, sort of augment the genome so that it includes those. And once that's done, we can now align everything again, but using the standard scoring scheme. So this is the first example where we didn't have the right scoring scheme to begin with, so we augmented the genome, and now the scoring scheme is appropriate. So we can go back and align everything. The other example I wanted to bring up was graph alignment. So this is a more recent phenomenon, but it's also a very popular thing to study. And if you haven't seen graph alignment, I can explain it purely in terms of today's thesis, which is graph alignment is basically dodging the fact that our scoring schemes don't understand anything about what non-reference alleles are present in the population. So it can't calibrate its penalties according to what alleles are actually present in the population. So one way that it seeks to sort of get around this is it'll take some of those alleles that are present in the population and include them in the reference genome representation and include them in the index. So for example, this diagram that you see at the top, which is a figure from a paper that I think Christina Boucher is going to talk about a little bit later as well, has basically a sort of backbone which represents the reference sequence, and then it's got a few sort of diversions and auxiliary paths that correspond to alternate alleles that are present in the population. And these things can be formed and indexed and aligned to. And if you're careful about how you pick the alternate alleles to include in the graph, then you can actually improve alignment correctness. You can improve various alignment accuracy metrics to the point where you're kind of approaching the ideal of if you had had a personalized reference genome for this particular individual. So this can definitely help improve alignment. And there's just a little pitch because this process of selecting the variants to include is, I think, also a difficult problem. We have a preprint about that. But this is basically the diagram describing what I just said. You don't have to follow all the squares and arrows. I would just summarize it by saying, in a first phase, our job is to select which of the alternate alleles we want to put into the representation, and then index that representation. Thank you. And then in the second phase, we want to align to that representation, at which point we can more or less go to our standard bag of tricks, standard scoring functions, seed and extend, things like this. So the parallel I want to exhibit between these two regimes of spliced alignment shown down here and graph alignment shown on the top is that, again, we're sort of splitting the problem into two phases. And the purpose of the first phase is that we want to gather the information we need so that we can augment the reference genome so that it has the things already that are hard for us to score in it, so that in a second phase we can do standard alignment with standard scoring schemes. Okay, so the whole thesis of this talk is basically to look at this and sort of wonder, are we doing a good thing here? You know, is this sort of the way we want to solve this problem? And so I went faster than I thought. It's actually my final slide. So we saw two variations on read alignment, spliced and graphed alignment. And they both have this flavor of modifying the reference to work around the fact that typical heuristics and scoring functions aren't appropriate in these settings. And what I wanted to say about this is I think we should wonder whether this is a paradigm that we can or should improve on. One of the things I think that the paradigm is for better or worse, allowing us to get away with is that we don't have to write down the real objective function, right? So if we had to write down the real objective function, then we would have to think about things like intron length distributions and how convincing is it when we actually see GTAG versus, say, a non-canonical splice site versus maybe a splice site that's one base off from a canonical splice site. It's allowing us to get away with not writing that down. We haven't really written down what's the true scoring function when we know the allele frequencies of the variants involved, for example, in an alignment. And this has the downside that we never end up really characterizing what's the objective we're trying to optimize. It makes it more difficult to compare tools. It also makes it more difficult to characterize what the heuristics of the tools are doing, because you can't isolate the heuristics if you haven't even written down what's the objective that you're trying to solve with the algorithm. So if we could write it down, one thing we could do is we could implement the optimal, slow, heuristic-free algorithm, and then we could compare the heuristic-free algorithm to the algorithm that we have and ask questions like, what's it missing, right? Which is the fundamental question about what are the heuristics doing. So that's all I had. So I'm curious to see what you guys think.
Questions?
It's a nice talk. First step, you extract some subsequence from your reads and then try to predict the splicing junction first. So when the reads are, the subreads are very short, if you have multiple mapping locations on the reference, then how can you resolve that problem?
Yeah, so I think the way typical tools will work is they'll only call a junction if they're confident, if they see a pattern that really does look like a true splicing pattern. If the readlets are multi-mapping, obviously that muddies the pattern. So, I mean, I'm not trying to state what a tool should or should not do, but I think that typically what tools will do in that area is throw out the multi-mapping reads, not allow them to contribute to the splice junction call. Yeah. But that's a good example of a problem that if we wrote down the true objective and we solved it using, say, heuristic-free dynamic programming, that would be a good example of the kind of heuristic we've now avoided, right? And we can actually start to study, you know, what is the effect of chopping things up into too small pieces so that things get too ambiguous, for example.
Yeah, I feel like I finally understand what your programs are doing after this talk.
Wow. In 7 minutes. Whoa. I hope you were recording that. Yeah.
So that aside, I think it's a great idea now that we have already annotated all these junctions and we kind of have an idea. So can we go back and parameterize the scoring function?
Yeah.
Like
absolutely.
Blossom 62 was doing, right?
Sure.
You have the log likelihood.
And another thing we now have is spliced alignments from many, many, many public RNA sequencing data sets, right? So there's repositories like Recount and things like that, where if we wanted to sort of take spliced alignment and bring in some of that graph alignment magic where we can actually estimate splicing frequencies in the population, we can bring that in as well.
Really get it right.
Yeah. Yeah.
Yes.
So when you have a problem, a quality score which says the probability that the alignment is correct, it seems like that probability can be based on prior information, like the distribution of indel length, which is when you introduce the prior information, you're also then biasing your alignment towards not detecting something that's an outlier.
That's true.
Versus you have probability just based on non-prior information, like the sequence of the read, where it's multi-mapping, things like that. Is that something people think about, like just separating out maybe these two types of quality score ideas?
Yeah, I mean, I don't know how that would fall out if people went farther down this road. Like, for example, with the spliced alignment problem, it's hard to imagine that you can write down a satisfying scoring function or a model that doesn't require some training, right? So like, if you're gonna say, okay, well, I need to respect the intron length distribution, well, you gotta say what that distribution is somehow, right? So I think if you go far enough down the road of, as I'm suggesting, actually writing down what the objectives are, at some point you're gonna inject summary statistics at least from known data. But I mean, as for graph alignment, perhaps, you know, again, you're going to need summary statistics for things like allele frequencies. I think that the way mapping qualities are typically calculated, again, is very tied to the scoring scheme. You know, the aligners search for high scoring alignments. They find one, maybe they find a second one, and now they've got the crucial features they need to calculate mapping quality, which is basically what's the distance between the best and the second best alignment score that I found. So that could be improved by including prior information, and I don't know exactly how you would think about whether you should or should not for the purpose of calculating mapping quality. But that's another really understudied area, I would say.
I think we ran out of time. Yeah. Let's thank our speaker again.